Welcome back to the deep dive. Today we are really getting into the weeds with a fascinating area of genetics. We're tackling Chapter 7 from Principles of Genetics, all about linkage, crossing over, and crucially, chromosome mapping in eukaryotes. Our goal here is pretty straightforward. Take this, sometimes pretty dense chapter material and make it stick. We want to really understand how genes are laid out on chromosomes and, maybe more importantly, how scientists first figured out how to measure the distances between them using, well, mostly just observation and math.
Absolutely. This is where genetics takes a really big step beyond Mendel's neat picture of independent assortment. We're looking at why certain traits seem to be inherited together, that's linkage, and then the mechanism crossing over that actually breaks those links sometimes.
And the story behind the first chromosome map is just incredible, isn't it? Alfred Sturtevant, as an undergrad
Right.
in 1911.
Pulling an all-nighter, apparently, with fruit fly data.
Yeah,
You didn't see
yeah.
the genes, you just inferred how often they separated. It's kind of mind-blowing. It truly was revolutionary. He basically said, look, the frequency of separation tells us the relative distance. It laid the groundwork for, well, everything that followed in gene mapping.
Okay, so let's break it down. Foundational concepts first. What's the core difference we need to grasp between linkage and recombination?
So linkage is pretty intuitive. Genes on the same chromosome, they're linked, physically connected. They tend to travel as a single unit during meiosis.
Like passengers on the same train.
Exactly. And recombination, that's the process that allows some passengers to switch trains, metaphorically speaking. It breaks the linkage and creates new combinations of alleles on a chromosome.
And the first hints that Mendel's second law wasn't the whole story came early on with Bateson and Punnett.
Yes, their work with sweet peas, flower color and pollen length. They did the classic dihybrid cross.
Crossing dominant for both, with recessive for both, getting the F1, then self-fertilizing that F1.
Right. And when they looked at the F2 generation, the numbers were just wrong. They didn't get that clean 9.3.3.1 ratio you'd expect for independent assortment.
What did they see instead?
The parental combinations, the original pairings of traits, were way, way overrepresented. It was a huge signal that these genes weren't assorting independently. They're physically stuck together most of the time.
Okay, so they're linked. But how tightly? How do we measure that stuck togetherness?
That's where the recombination frequency, or RF, comes in. The key is using a test cross. You take your individual heterozygous for both linked genes and cross it with one that's homozygous recessive for both.
Why the test cross specifically?
Because the recessive parent only contributes recessive alleles, so whatever alleles the offspring gets from the heterozygous parent directly determine its phenotype. It makes counting easy.
Ah, okay. So you just count the offspring.
You count the proportion of offspring that show new combinations of traits, the recombinant types. If, say, 80 out of 1,000 offspring are recombinants, your recombination frequency is 80 divided by 1,000, or 0.08, 8%.
And that percentage directly reflects how often recombination happened between those genes during meiosis.
Precisely. And there's a fundamental limit here. The recombination frequency between any two genes can never be more than 50%.
Why 50%?
Because 50% recombination is what you get when genes are assorting independently, either because they're on different chromosomes altogether or just so incredibly far apart on the same chromosome that a crossover between them is pretty much guaranteed.
So anything less than 50% recombination frequency.
That's your proof of linkage. The lower the frequency, the tighter the linkage, the closer the genes are on the chromosome.
Got it. Let's shift from the numbers to the biology. What's physically happening in the cell during recombination? You mentioned crossing over.
Right. Crossing over is the actual physical exchange of segments between homologous chromosomes. It's a very specific event during meiosis I.
In prophase, right? When the chromosomes have duplicated and paired up.
Exactly. You have that structure called a tetrad, 4 chromatids all together, 2 homologous chromosomes, each already duplicated.
And the exchange happens between all 4.
No, and this is key. A single crossover event only involves 2 of the 4 chromatids, one from each homologous chromosome.
Okay, so 2 chromatids swap pieces.
Yes, and the result, after meiosis is complete, is that out of the 4 resulting chromatids, which end up in the gametes, 2 will be recombinant, carrying new allele combinations, and 2 will remain parental, unchanged.
How does the cell actually do that swapping? Is it just random breakage?
Oh no, it's a very precise enzyme-mediated process. Think of it like molecular surgery-specific enzymes break the DNA strands on the 2 chromatids and then repair them, but in a crisscross way. joining the fragment from one to the other.
And later in Prophase I, we see these structures called chiasmata.
Yes, the chiasma singular, chiasmata plural. These are the visible points of connection, the crosses that we see between the homologous chromosomes as they start to condense. They're thought to be the cytological manifestation, the leftover entanglement from that earlier physical exchange.
And these chiasmata aren't just signs of recombination, they're structurally important too.
Absolutely vital. They physically hold the homologous chromosomes together until anaphase the first. Without chiasmata, the chromosomes might drift apart prematurely, leading to errors in segregation, like aneuploidy, the wrong number of chromosomes.
Okay, that makes sense. Now, the really elegant proof connecting this physical crossing over with the genetic recombination we measure came from Creighton and McClintock.
Yes, Barbara McClintock and Harriet Creighton in 1931, working with maize with corn. It's a landmark experiment.
What was so clever about it?
They found a strain of maize that had a chromosome 9 that was visibly different under the microscope. It wasn't just a normal chromosome 9.
How was it different?
One end had this big, dense blob called a heterochromatic knob, and the other end had a piece of a different chromosome attached, a translocation.
Yeah.
So they had visual markers on the chromosome itself.
And they also tracked genetic markers on that chromosome.
Exactly. They tracked genes for kernel color, CC, and kernel texture, WX, WX, that were known to be on chromosome 9.
So they could follow both the genes and the physical chromosome markers at the same time.
Precisely. And they showed a perfect correlation. Whenever they observed genetic recombination between the kernel color and texture genes in the offspring,
Let me guess. They saw.
they saw that the physical markers, the knob and the translocated piece, had also swapped between the homologous chromosomes. Genetic recombination mirrored the physical exchange of chromosome segments.
Wow. So that sealed the deal. Genetic distance really is about physical exchange.
It absolutely did, and it validated Sturtevant's original idea that recombination frequency could be used to map genes. This led directly to the concept of the genetic map distance.
Measured in centiMorgans?
Yes, centiMorgans, abbreviated CM, in honor of T.H. Morgan, Sturtevant's mentor. One centimorgan, or one map unit, is defined as the distance between genes for which you expect 1% recombination frequency.
So if we do a simple two-point test cross, like mapping vestigial wings, EAG, and blackbody B in fruit flies, let's say figure 7.11 in the text.
Okay.
You cross a VGB plus fly with a VGB, VGB fly, you count the progeny, you find, say, 180 recombinants out of 1,000 total offspring.
Right. So 180 divided by 1,000 gives you a recombination frequency of 0.18, or 18%.
Which means the distance is.
Approximately 18 centimorgans. For shorter distances, the recombination frequency is a pretty good estimate of the map distance.
But you mentioned it gets trickier for longer distances. Why?
Because of double crossovers. If two genes are far apart, it's possible for two crossover events to occur between them in the same meiosis.
And what does a double crossover do to our count?
Well, think about it. The first crossover swaps the alleles, creating a recombinant chromatid. But if a second crossover happens between the same two chromatids further down, it swaps them back again.
Ah, so the final chromatid looks parental, even though two crossovers happened.
Exactly. So a simple two-point cross underestimates the true distance for genes that are far apart because it misses these double crossovers.
Which brings us to the power of the three-point test cross, mapping 3 linked genes at once.
This is the gold standard for accurate mapping and, crucially, for determining the order of the genes on the chromosome.
How does mapping 3 genes help find the order? Let's use the example from Bridges and Ulbrecht CEC-CV genes in Drosophila. You do the test cross, you get 8 different phenotypic classes of offspring.
Right, because there are 3 genes, each with 2 alleles. And the key insight is to look at the rarest pair of phenotypic classes among those 8.
Why the rarest?
Because those represent the double crossover DCO events. For a double crossover to occur, you need 2 separate exchange events, one in the interval between the first and second gene, and one in the interval between the second and third gene. That's inherently less likely than a single crossover in either interval.
Okay, so find the 2 rarest counts. Those are the DCOs. How does that tell us the gene order?
You compare the allele combination in the DCO offspring to the allele combination in the original non-recombinant parental offspring, which are the most frequent classes. And you'll see that the DCO classes look just like the parental classes, except for the allele of the gene that's located in the middle. That middle gene's allele will be flipped relative to the alleles of the 2 flanking genes.
So if the parent was AB cap and the DCO is AB cap BC,
Then B must be the gene in the middle. The double crossover event effectively swaps only the middle marker relative to the outside markers. It's unambiguous.
That's really clever. So the DCOs give you the gene order directly?
Instantly. Then, to calculate the map distances between the genes, say between A and B, you sum up all the single crossovers that occurred in that AB interval, plus all the double crossovers. Because remember, every double crossover event includes one crossover in the first interval, AB, and one in the second interval, BC. You have to count them for both intervals.
Right, that makes sense. You can't forget the DCOs when calculating interval distances.
Never. Otherwise, you'd still be underestimating the distances.
The three-point cross also reveals another phenomenon, doesn't it? Something called interference.
Yes, interference, usually denoted by I. It's the observation that a crossover event in one region of a chromosome tends to, well, interfere with the probability of a second crossover event occurring nearby.
So crossovers aren't completely independent events along the chromosome's length.
Not entirely, especially over shorter distances. One crossover seems to make the nearby chromatin less receptive, somehow, to undergoing another exchange.
How do we measure this interference?
We first calculate the expected frequency of double crossovers. If the crossovers in the two intervals were totally independent, we just multiply the recombination frequencies for each interval. For example, if interval 1 has 10% recombination, 0.1, and interval 2 has 20%, 0.2, we'd expect 0.1, 0.2 equals 0.02, or 2% double crossovers.
Okay, that's the expected frequency.
Then we compare that to the observed frequency of double crossovers, the actual proportion of DCO offspring we counted in our experiment.
Yeah.
The ratio of observed DCOs to expected DCOs is called the coefficient of coincidence, C.
Observed over expected. And interferences.
Interference I is simply 1 minus C. So if C is 0.6, meaning we only saw 60% of the expected double crossovers, then interference I is 1.6 equals 0.4, or 40%.
What does a high interference value mean? Like close to 1?
I interference close to 1 means C is close to 0, meaning we observed almost no double crossovers compared to what was expected. The first crossover strongly inhibited the second one. This is typical for genes that are close together, say less than 20 centimeters apart.
Okay. So we've built this genetic map, measured in centiMorgans, showing relative gene order and distances based on recombination. But how do we connect this abstract map to the actual physical chromosome we see under the microscope?
That's where cytogenetic mapping comes in. It aims to locate genes relative to physical cytological landmarks on the chromosome, like the banding patterns you see after specific staining techniques or other structural features.
It's about finding the gene's physical address, not just its neighbors.
Exactly. One classic technique, especially powerful in Drosophila, is deletion mapping.
How does that work? Let's use the example of the white-eye gene, W. It's recessive.
Okay, so you have flies that are heterozygous, carrying one wild-type allele, W+, and one recessive white allele, W. Normally they have red eyes because W+ is dominant.
Right.
Now, suppose you cross these flies to flies that carry a chromosome with a known physical deletion. A segment of the chromosome is actually missing. We call this a deficiency, or DF.
So the offspring inherit one normal chromosome, potentially with W, and one chromosome with a piece missing, the DF chromosome.
Yes. And here's the test. If an offspring that inherits the W allele on the normal chromosome and the deletion chromosome shows the white-eyed phenotype,
Wait, even though it should have inherited a W+ allele from the other parent? Oh, unless
unless the wild-type W+ allele was located within the piece of chromosome that got deleted on the DF chromosome, the deletion has uncovered the recessive W allele, allowing its phenotype to be expressed.
So if a specific deletion uncovers the recessive phenotype, the gene must lie within the boundaries of that deletion on the physical chromosome.
Precisely. You can test a series of deletions with different known endpoints to pinpoint the gene's location quite accurately. There's also duplication mapping, which is the opposite logic. A duplication covering the gene restores the wild-type phenotype.
This brings up a really important point mentioned in the text. Genetic maps and physical maps are collinear but not proportional. What does that mean?
Collinear means the order of genes is the same on both maps. If the genetic map says ABC, the physical map will also show ABC in that order.
Okay, same order, but not proportional.
Not proportional means the distances aren't scaled the same way. A region that looks short on the genetic map, meaning few crossovers happen there, might be physically very large in terms of DNA-based pairs.
And where do we see that, typically?
Most notably around the centromeres and the telomeres, the ends of chromosomes. These regions tend to have very low rates of crossing over. So genetically, they appear condensed genes there, seem very tightly linked. with small centimorgan distances between them.
But physically, they could be huge stretches of DNA.
Exactly. Conversely, other regions might be recombination hotspots, where crossing over is frequent. These look expanded on the genetic map compared to their actual physical size. The relationship is linear in order, but distorted in scale.
Okay, let's bring this closer to home. What about mapping genes in humans? We obviously can't do controlled test crosses.
No, that presents significant challenges. Human geneticists have to rely on analyzing existing family pedigrees, which are often small and don't have the ideal structure for mapping.
So how did they first establish linkage in humans?
One of the earliest successes was demonstrating linkage between the gene for the ABO blood groups and the gene for a rare dominant disorder called Nail-Patella syndrome, or NPS1.
How did they do that with just pedigrees?
By carefully analyzing large, informative pedigrees where both traits were segregating, they looked for non-random associations between specific blood types and the presence or absence of the syndrome across generations. Using statistical methods, they could estimate the recombination frequency.
And they found linkage.
Yes, they estimated the distance to be around 10 centiMorgans and eventually pinpointed both loci to near the tip of the long arm of chromosome 9.
But progress was slow, relying just on visible traits or diseases.
Right. Very slow. The real breakthrough for human mapping came with the discovery and use of molecular markers. These are variations in the DNA sequence itself, like SNPs, single nucleotide polymorphisms, or RFLPs, restriction fragment length polymorphisms, that are common in the population.
How do these help?
Because there are millions of these markers scattered throughout the genome. Even if you don't know where the disease gene is, you can test for linkage between the disease phenotype in a family and hundreds or thousands of these anonymous DNA markers whose locations are roughly known.
So you're looking for a marker that tends to be co-inherited with the disease?
Exactly. If a specific marker allele is almost always found in affected individuals in a family, and rarely in unaffected ones, that marker must be physically close to, and therefore linked to, the disease gene. This is how the gene for Huntington's disease was famously mapped to chromosome 4, long before the gene itself was identified.
That really revolutionized human genetics.
It absolutely did. It paved the way for the Human Genome Project and for identifying genes for countless inherited conditions.
Finally, let's zoom out. What's the big picture evolutionary significance of all this linkage and recombination?
Well, recombination is fundamentally important for evolution. By shuffling alleles between homologous chromosomes, it creates new combinations of existing genetic variation.
Why is creating new combinations so important?
Because it allows natural selection to act more efficiently. Imagine two different beneficial mutations arise independently on different copies of a chromosome in a population. Without recombination, it's very hard to get both beneficial alleles onto the same chromosome to be passed on together.
But recombination can bring them together.
Yes. Recombination can create that doubly advantageous chromosome much faster than waiting for a second mutation to occur on a chromosome that already has the first one. It speeds up adaptation.
And you mentioned earlier that chromosome inversions can suppress recombination.
That's right. If an individual is heterozygous for an inversion, meaning one chromosome has a segment flipped relative to the other, crossing over within that inverted loop during meiosis leads to big problems.
What kind of problem?
You get structurally abnormal chromatids. One might end up with two centromeres, a dicentric braid which gets torn apart during anaphase, and another might end up with no centromere, an eccentric fragment, which gets lost.
So the gametes containing these recombinant chromatids are
Usually inviable. The only viable offspring tend to be those that inherited the non-recombinant parental chromosomes. So effectively, recombination between genes within the inversion is suppressed in the offspring.
And this is used experimentally.
Oh yes, geneticists use balancer chromosomes with multiple inversions specifically to prevent recombination and keep desired combinations of alleles together generation after generation. And it's also thought to be a key mechanism in the evolution of sex chromosomes, like the X and Y. The Y chromosome has several large inversions relative to the X, which essentially shut down recombination between them in those regions, allowing them to diverge significantly.
Creating those distinct evolutionary strata mentioned in the text. Fascinating.
It really is. It shows how chromosome structure itself influences evolution.
Okay, let's try to wrap this up. Key takeaways from this deep dive into linkage and mapping.
Well, first, linked genes don't assort independently. They violate Mendel's second law.
Right. And the frequency of recombination between linked genes gives us a measure of their distance in centiMorgans.
The physical basis for that recombination is crossing over between homologous chromosomes in Prophase I, visible later as chiasmata.
And the three-point test cross is the powerful tool that lets us determine gene order using those rare double crossover classes.
Don't forget interference, the fact that one crossover can inhibit another nearby.
And finally, cytogenetic mapping anchors our genetic map to the physical chromosome, revealing that while gene order is conserved, collinearity, the genetic distances aren't directly proportional to physical distances due to uneven recombination rates.
A pretty comprehensive tour. And maybe a final thought for you to ponder, connecting back to the mechanics. We stressed how crucial crossing over and chiasmata are for proper chromosome segregation and meiosis, preventing aneuploidy. But, interestingly, in some organisms, like male Drosophila fruit flies, there is essentially no crossing over during meiosis.
No crossing over at all in males. How do their chromosomes segregate correctly then?
Exactly. Given that lack of chiasmata to hold homologs together, what alternative mechanisms might have evolved in male flies or similar organisms to ensure their homologous chromosomes still find each other and separate accurately during meiosis? Something to think about.
That is a great question to mull over. It highlights that there's always more complexity. Thank you so much for guiding us through this chapter.
My pleasure. It's fundamental stuff.
And thank you, our listener, for joining this deep dive. We hope breaking down linkage, crossing over, and mapping helps you connect these critical concepts. From the Last Minute Lecture team, thanks for tuning in.